Dr. Jithendra Panneerselvam is a senior lecturer in the Department of Pharmaceutical Technology at International Medical University, Kuala Lumpur, Malaysia. His expertise in tissue engineering, biomaterials, wound healing, nanoformulation. He has also secured various internal research grants and publishing several research and review articles in reputed journals. We welcome Dr. Jithendra Panneerselvam to enlighten us with the worthwhile insights. Over to you, sir.
Right. Thank you very much indeed. Can you hear me?
Yes, sir. It's clear.
Right. So just hang on a minute. Allow me to share my screen. Right. Just a minute. Just hang on a minute. Right. Right. Well, good morning.
Sir, it's get mute.
Oh, yeah. Sorry. Haven't started yet, so thank you very much for your kind introduction. I'm gladful to be part of this event. I thank the organizer, Dr. Purushothaman, and other teams who made this event quite successful and, of course, made this event more useful for the young generation. Well, I take this opportunity to extend my thankfulness to them because I'm going to share my experience that certainly I believe it's going to be useful for the young generation, which may have an idea to pursue a research as a career. Right? Just I'm going to share. Right? Can you see my screen now? Can you confirm?
No, sir. It's not visible.
All right, just a minute. Yeah, I haven't shared it yet. Right. I think now I guess now you can able to see it. Can you?
Yes, sir. It's clear.
Right. Thank you. Okay, right. Here you are. So, very good morning again. So here, good afternoon in Malaysia and morning in India. So I'm going to present a topic on fundamentals of tissue engineering and regenerative medicine. It sounds a bit sophisticated term, but it's quite used and it's been there for centuries. Okay, so I'll put the session in a very simple way so that the students who are the participants who can able to grab the theory behind it, which would be useful for them to pursue the career in tissue engineering and regenerative medicine. So first of all, the disclaimer slide. I represent only myself and not from my university. The opinions and views which is going to share by myself is purely my experience. No way it is relevant to the IMU. And I'm going to deliver the content is as accurate as possible to the best of my knowledge. Well, let me talk about the introduction part, as you are quite excited to know about what is tissue engineering and regenerative medicine, isn't it? So basically, tissue engineering and regenerative medicines are quite interlinked together, and often they spelled and interchangeably. So in fact, tissue engineering is a part of a tissue regenerative medicine. As the name sounds, as you can see, regenerative medicine. So you can split the term and you can at least get the point what it is all about. So something you try to regenerate, particularly in organ or tissue, which already having an issue or disease condition or malfunction conditions, right? So you want to replace it completely or partially in order to restore the function of an organ. So specifically, that's the one of the reasons this, the field of interest has been originated in originally. But when you look at the history, it's been there since from 1000 BC, if I'm not wrong, and somewhere they will keep doing the experiments or transplanting the organs and to regenerate the malfunction organs. But officially, when you look at it, when the term the tissue engineering was officially recorded only in the late 19th centuries. So if it'll start more, it attracted more attention in the 1980s. From then onwards, the journey started. So this field, basically, it is amalgamation of both the biology and engineering. And the objective is to restore and maintain and improve the functions of any defective organ. All right. In fact, it is going to be one of the hot selling cake field where they're focusing on alternative for organ donors. As you all know, the donation of the organ is quite limited, even though we have billions of populations, but we don't have so many people for volunteering to donate the organs. It's quite fact. Even an expertise, even a well-educated, they are reluctant to do so, right? So is there any way of solution to solve this issue? So in that mind, the regenerative medicine pops in it. Well, I'm going to show you here, the pictures are here, or artificially synthesized in the laboratory scale. So this is a bladder, urinary bladder, trachea, and heart, even earlobe. You might be wondering what is about to look like a lollipop, but it is a kidney. Okay, so they made it different attempts. They tried to partially or fully, to organize or to evaluate a function of an organ. So this is the triggering point where they find a solution for any defective organs. Right. Let's talk about the tissue engineering triad. So we have talked about the examples, see that the urinary bladder, trachea, heart, earlobe, etc., etc. We have seen in the last slide, right? So how are they going to do it? So they can do it with the help of three important elements. So what are they? They are biomaterials, and of course signaling molecules and cells. So that's the reason they call it a triad. So incorporation or participation of these three elements is going to be the successful entity of tissue engineering. If you want to regenerate any of the organ or tissue, you need to have a role of these three important elements. We'll be seeing in detail about it. Right, this is a schematic representation of how the tissue engineering is being processed. So for a simple way of representation, as you can see, you can take the cells, culture outside, proliferate the cells, and of course you can put it in the scaffold materials. Scaffold materials, you might be wondering what it is all about. It is a supportive material in which the cells will sit and proliferate, and of course it does express its entirety. Then you can culture in the in vitro condition and you can transplant it. It sounds simple, but it's quite tedious process. Even though I have said about it more than a century, more than 1000 BC, it's there in the practice, but on and off, it was not properly recorded and it was not properly researched. From the late 19th century, the scientific improvement will lead to the attention of the tissue engineering technology in the regenerative medicine. Right. So out of the three triad materials, let's start with the cells. In a simple manner, cells we all know it's a unit of living being, isn't it? Anything which is made up of cells, we call it as living being. So here every cell does its own function, right? So for instance, if you're going to take the skins are made up of fibroblast cells. If you're going to take cartilage material, it is going to be secreted by the chondrocyte cells. Similarly, bone by bone cells or osteoblast, myocytes like heart cells, adipocytes, all right, for fat cells. So the list goes all along, okay? But whatever the organ you want to regenerate or whatever the tissue you want to regenerate, the choice of the cells depends on it. For instance, if you want to regenerate a partial heart, then you need to culture the myocytes. You should not go and culture the fibroblast, right? So you choose the right choice of the cells to meet your objective to regenerate a particular organ or tissue, right? Right, so let's talk about the types of cells. Anyway, I'm just briefly going to present it. So what are the different types of cells you might have studied even in your schools, right? Or if you're already pursuing your undergraduate, perhaps when you're on the science field, you might have studied. It's not a new term which you're gonna read now. So autologous, of course, anything the cells which harvested from the same person, we call it autologous. Anything from the same species, we call it allogenic cells. Right? Perhaps a person donates his cell to another person, right? It's the same species. Xenogenic, of course, from different species. From animals, from animal kind, they might be taking a cells from humans. So when you transplant the cells, we call it as xenotransplant. When you harvest the cells for transplantation, we call it as a xenogenetic cells. Isogenic, of course, this is from iso means same, from twins. If they have twins, and they can donate the cells from one twin to another one. So that we call it as isogenic cells. Primary cells, basically are cells which freshly harvested from a tissue, an organ, we call it as cells. When you do the proliferation, we call it a passage in a technical term. When you keep passaging the primary cells, then the lineages, the cell lineages, we call it as secondary cells. And of course, the most important and the very hot topics, okay, even not now, even for the past 2 decades, it's been there for stem cells, right? Plays a significant role in the regenerative medicine. You don't have to, for other things, of course, you may have to depend on fully differentiated cells in order to achieve a partially or fully of an organ. But stem cells, you don't have to worry. From single stem cells, you can differentiate it with the proper signaling to any type of cells. Right, so in the stem cells, again, I'm going to glimpse of the stem cells. It's a vast ocean. You have to go and read a lot about it if you're quite interested in that. So we learn about the stem cells, multipotent stem cells. These are neural stem cells, mesenchymal hematopoietic stem cells. And in terms of pluripotent stem cells, it will be classified embryonic stem cells and perinatal stem cells and pluripotent stem cells, induced one. So out of these, all, of course, the stem cell, as I said before, it's the most attractive field in the regenerative medicine. You might have seen it, the advertisements, in the science, even hospitals, right? They might have asked you to preserve your umbilical cord stem cells that can be useful for any ailment when a person grows, during a lifetime, her or his lifetime, right? So the rest of the cells, of course, it has to be in a, can be used it. The most versatile is going to be, the embryonic stem cells and, of course, pluripotent stem cells. But again, due to the ethical reasons, the research on embryonic stem cells are quite almost nil, right? They are here and there. It's being done, but that's in the animal model. Of course, it's not been ethically approved. But again, what's going to be the alternative stem cells? The induced pluripotent stem cells is going to be quite behave like embryonic stem cells, but in fact, it is derived from a somatic cells. So you can have a more explorations about a lot. Even a gene therapy has been done by induced pluripotent stem cells. There's a company if I'm not mistaken, a company in Bangalore. They do extensive work in the iPSC cells. I think it's, if I'm not mistaken, it's an INSTEM. Right, so a lot of work is going on, especially to regenerate, to give a solution. Especially to give a solution, it might be the problem might be from genetically oriented or due to the trauma or accident or trauma cases that might have originated. So that could be the potential source for replacement. Right, this is another schematic representation of the cell source. As I said about different types of cells, right? We have listed about it. Again, so if you want to take up the differentiated cell, of course, when you harvest the cell from a brain, we call it neural cells. If you harvest from a brain, if you're from your heart, myocytes, if you're hepatocytes from kidney, from your liver, and of course, similarly for osteocytes from bone, chondrocytes from cartilage material. So wherever the cells you've been harvesting, the cell's going to differentiate into that type of organ. All right, so if you're interested is about the bone cell, harvest the cells from the bone and start culturing in an in vitro condition. And of course, as I said, one of the triad material, of course, still you need to have the triggering factor, which is biomolecules, also called the signal molecules, that is going to be ensured it is going to be differentiated into your interest of cells. Okay, so that can be done. And of course, you can culture in the in vitro, and with the help of the scaffold material, you can do it in a 3D structure and you can implant it back. Okay, this is just a glimpse of information about the cells. So we talked about one of the elements, which is cells, which is a brief enough for you to explore more about it. Because studying a cell itself, a lot of a lot of fields are there. Biological science people, you can take over that and explore more information on the cells. Let's talk about scaffolds. Scaffolds looks quite odd for you. What's the term? When you look at the dictionary, scaffold is nothing but a supporting material, three-dimensional material. A simple way of understanding, as you could have observed, when people are constructing any building, right? First of all, they erect the scaffold. Scaffold is a three-dimensional structure where it offers a place where the people are going to stand on it and erect the building. So what will they do? Once they constructed the building, are they going to retain the scaffolding? Not at all, isn't it? So once they complete the construction process, they're going to dismantle it. The same way here, the purpose of the scaffold is it allows the cells to grow on it, secrete its secretions, okay, and give architecture. All right, it gives a three-dimensional architecture for the cells to grow on it. So that's the beauty behavior of the cells are when the cells are grown on a monolayer in a tissue culture flask, it behaves differently. But when you try to grow the same cells in a three-dimensional architecture, it behaves quite similar to your actual in vivo condition, even though it doesn't meet exact in vivo condition, but it quite meet quite near to the in vivo condition. So the most of the studies in vitro modeling, they prefer on 3D culturing. So in that way, the scaffold is going to play an important role. It's going to be a supportive material on which the cells will be settled down, proliferate, and achieve its unique structure, and later is going to establish its functions too. Right. Properties. So how the properties, as I said, biocompatibility. What does it mean? So it should be compatible with the physiology, right? When something is going to be injected or implanted in a person, the gift of our life is the immune system, right? Anything which embedded into your body, immediately your immune system will take it off and try to expel it. Okay, that's the beauty of it. But in this case, this immune system itself going to be quite a villain for us, especially for tissue engineering. Anything you're going to implant it, of course, it try to reject it, right? If it rejects, of course, the whole objective of exercise is going to be dumped. So in this case, choose a material which should be compatible to the physiology, right? So that's very important. Second one, biodegradability. As I told you before, upon completion of any building, you need to dismantle the scaffold, right? Similarly here, once the cells has been grown, you need to remove the scaffold. How can you remove the scaffold? You can't remove it, isn't it? It's not like you're building scaffold. So here, automatically, it has to undergo degradation process. That's called biodegradability. So the moment it degrades, that void space will be filled by the cells. Okay, so a good scaffold material or biomaterial should be biodegradable in nature. Mechanical support, right? This is a very important element. For instance, different organs requirement different mechanical strength. For an example, if you're going to harvest a skin cells or artificial skin, where you need a support, a bit of elastic in nature, and it should be flexible in nature too, right? And at the same time, if you're going to replace a malfunction or broken bone, the broken bone need not require any flexibility. It needs a rigidity, right? So in this case, you need to choose a material which gives a support in rigid manner so that the bone cells will grow on it. All right, so mechanical property is very important. Choose the mechanical property relevant for your application. Accordingly, you need to choose it. Porous structure, right, very important, because you can see is a good example. The cells can migrate into the porous, because if you grow on the surface, as I said, it's a three-dimensional, right? The cells has to migrate inside. So if they want to migrate inside, of course, there must be a pore size quite bigger than the cell size, right? So it must have a porous that will allow the cells to migrate inside and proliferate. At the same time, it will also allow the nutrients to migrate, and nutrients and oxygen transfer will move inside in depth of the composite and allow these cells to respirate and proliferate. So pore structure plays a significant role. Again, if the pore structures are quite bigger in size, that may affect the mechanical property. So you need to strike a balance between the pore size and of course the mechanical property, right? So those are quite technical terms. You have to be in focus when you develop any biomaterials. Another thing, architecture. Not all the materials can be molded according to your requirement. For instance here, so one of the polymers they've used, this is the hip bone. They can mold it according to the shape they wanted, right? That gives them more flexibility, especially for the regeneration purpose. So if a biomaterial could be good at all the properties, but it can't be architectured a particular mold, then it's going to be useless, right? The same way, a good scaffold material or biomaterial is supposed to be having these properties in order to call it as an ideal biomaterial for making scaffolds. Again, it's a definition. Biomaterials are substances which are going to interact with your biological system. We call it as biomaterials. Okay, so again, it has the choice of the biomaterial, as I said, would vary, depends upon your objective. Okay, and very importantly, as I said, the biocompatible earlier, right? So that is also very important, because if your system, your immune system can able to recognize it's a foreign substances, then immediately that it will going to be rejection. Right, I'm going to give a brief introduction about the classification of biomaterials. Broadly, it's been classified into two types, natural and synthetics. So naturals, as you can see, protein-based, polysaccharide-based, this goes on. And synthetic, it goes on polymer, biomaterial, polymer-based as well as ceramic-based. Right, both the natural and synthetic materials has both the pros and cons. If you take the biomaterial, especially natural origin, might be good at biodegradability, but may be poor in mechanical properties. Okay, where this may be a good at mechanical property, perhaps may be poor in degradability. So that's a good one example I'm just pointing it. So when you take the biomaterials, of course, it's naturally available. So most of the time, the immune rejections can be minimized. I can't say it's completely removed it, but you can minimize the immune rejections. But it can be manageable with the help of the immunosuppressants. Whereas a synthetic, of course, this is a new xenobiotic, which means new substances entering into the body. Your immune system will immediately recognize and try to expel it. Right? So, as I said, both has the pros and cons. But again, nowadays, there is a combination of the natural and the synthetic composites forms. So the composites which can able to give the properties of the both natural and of course synthetic, which going to be considered as one of the ideal requirement for any tissue engineering process.